Hi guys, in this video we'll be looking at exam style questions for the microscopy practical.
Welcome back to our videos for microscopy. We've covered a lot of stuff so far about the background of what microscopes are designed for, and then how we use them, how we prepare slides, how we would analyse the slides, and the different evaluations and conclusions that we can make through the experiment. Now what we need to do is tackle some exam style questions that cover each of these sections and the sort of thing that you might see in your exam. So this first video will be talking mainly about the planning and the implementation, and then the next questions will be covering more of the analysis and evaluation side of things. So let's read the first question here. So it says that a researcher is examining a crop which may be exposed to a recent bacterial endemic. The bacteria are thought to invade the epidermis of the leaves within the crop. Part A says, describe a method the researcher could use to prepare the epidermis for inspection under a light microscope, and it's about 4 marks. Before I show you what I've written, let's just analyse the question here. So basically we've got a crop and there's a potential bacterial infection in the epidermis of the leaf, and he wants to use a light microscope to examine the epidermis and inspect it, and we want to describe a method. So when it says describe, look at the command word. That is basically going to be a situation where you need to list each step in the preparation. It's not really an explanation or a suggest. This is just some sort of method that you should really be familiar with. And it's referring to back in the planning section where we talked about how we would take an epidermis and prepare it for a slide. So let's look at what I've written here. So the first thing I've said is gather a fresh sample of leaves from several of the crops. So just writing the idea that you've got the leaves from several samples or several crops is a good start. And taking different samples is good because it covers a range of plants where some might be infected and some might not. So we've covered a good ground there. We then tear the leaves and then use the forceps to peel the epidermis cleanly away. So mentioning forceps and peeling the epidermis off would be another mark here. We then place the epidermis onto a clean slide, onto a drop of water, and then you place the cover slide upon this using a mounted needle. Again, this is all about what we talked about in implementation. We use forceps, mounted needles, and also gloves, which I haven't mentioned here, to reduce the contamination. You don't need to go into why we do this because it's just describing the method question. And then I put at the end, wipe away any excess water and stain if necessary. So although there are about 5 separate marks going on here, in biology you'll find that a lot of questions, there's a list of marks that you're allowed to pick up and they're just looking as to which ones you've written down. It's never a bad idea to write down extra facts if you can think. I've covered the method up until wiping away the excess water. Staining isn't really required. We don't know much about what sort of stains this leaf would need, but it's just something to mention and it acknowledges the fact that you're aware of it. So that's how I would answer this question. The next question, part B, says, one disadvantage of the light microscopes is that their magnification is limited. State an instrument the researcher could use with higher magnification if the source of infection was a very small virus. So this kind of goes back to the background video that we did, and we mentioned that light microscopes do have limited magnification, but there are other instruments that have a higher power. And we know that certain things are visible to the naked eye, certain things are visible to the light microscopes, and certain things are visible to electron microscopes. A very small virus is only really going to be picked up as a large molecule underneath an electron microscope. So the answer that I've put for this question is simply an electron microscope. And you can put scanning or transmission, it doesn't really matter, but that's the mark for that one. Part C then says, in preparing the slide for the light microscope, what 2 actions could the researcher take to minimise contamination of the slide? Explain your reasoning for each. So this kind of follows on from part A, in that why did he do things in certain ways, or why would you prepare things in a certain way to reduce contamination? And it says to explain your reasoning for each. So the reason this is 4 marks is you would have your 2 actions, and for each action the reasoning for each one. So what I've written here is, the first thing I've said is to wear gloves when handling the epidermis and when handling the slide. So that's one thing you can do, that's one action you can take. So this is to prevent bacteria or other microbes on the hand from infecting the specimen, which may have shown up under the microscope. So then you've explained it. You've explained that it can be contaminated from your skin because your hand has a lot of bacteria living on it, good and bad bacteria, and you're deliberately trying to look in this situation for a leaf that's been infected by bacteria because of this endemic. You don't want to be looking at a leaf that's been infected from the bacteria from your hand because that will give you a false impression that something's going on. So explaining this is really important for each of your marks here. The second mark I said was to use forceps to transfer the epidermis. So this is not only to prevent germs, but also physical distortion, because of the grip of the hand is less precise, so it's more likely to get damaged if not using forceps. So again, using forceps and explaining why we do that. So that should cover your 4 marks. So these are just a few questions that we've covered based on the planning and the implementation of the method. In the next video, we'll discuss a few questions based on analysing specimens and evaluating what we've gathered. I'll see you in the next video.
Hey guys, I hope you enjoyed the video. If you're looking for an amazing A-level biology resource, join me today in my series of engaging bite-sized video tutorials. Just click the Snap Revise smiley face and together let's make A-level biology a walk in the park.